Absolutely.
Welcome, everyone. I am Toonie Shartner, the Executive Director of District Hall and Venture Cafe Providence. And we really enjoy partnering with Rhode Island Bio for this series, the Rhode Island Innovator series. So welcome, everyone, and I'll just hand it off to you, Carol.
Thank you, Toonie. Always great to partner with Toonie and Jenna and Amy and her team at the Venture Cafe. So thank you again for hosting us. So good morning, everyone, and welcome to our webinar, COVID-19 Vaccine Update. And it's about what we've learned and what it means for the future. So my name is Carol Malysz. I'm the Executive Director of Rhode Island Bio, which is Rhode Island's life sciences trade organization. And as the voice for the life sciences industry in Rhode Island, we provide education, collaboration, and advocacy. So in short, we help science advance so science can advance lives. I want to thank our other sponsors, Bio, Rhode INBRE, Driven, and everyone who has supported this event. Our co-host Bongsup Cho is in his car on the way to get a vaccine. So he's not going to be with us on screen, but I'll say a few words on his behalf. And Bongsup is the program director for Rhode INBRE, which is a statewide network designed to build the biomedical research capacity of Rhode Island institutions by supporting and developing talented individuals committed to research careers in Rhode Island. And the Rhode INBRE program was made possible through the Rhode Island Institutional Development Award Network of Biomedical Research Excellence and by the Driven Accelerator Hub, both funded by the National Institute of General Medical Sciences of the National Institutes of Health. We really appreciate the collaboration we've had with Rhode INBRE and Driven, and all of the support that Bongsup and his team have given to our Innovator series. So now it gives me great pleasure to introduce our moderator for today's session, Michael Elliott, Chief Scientific Officer at Immunovant. And previously, Michael served as Vice President, Immunology Scientific Innovation at Johnson & Johnson in Cambridge, with responsibility for external collaborations and partnerships in immunology. Dr. Elliott has held several various positions at Johnson & Johnson, including General Manager and Site Head, Transform Pharmaceuticals, and Senior Vice President of Immunology at Centocor, where he led the Clinical Development Group. Michael is also on our Rhode Island Bio Board of Directors. So welcome, Michael.
Thank you, Carol, for that introduction. It's great to be here. Thank you, Toonie, also. It's my pleasure today to chair this session, and I'm chairing it from my home office here in Rhode Island, where I've spent most of the last 12 months, and I'm getting a bit sick of my home office. So I'm really looking forward to being liberated from it by the COVID-19 vaccine, and that's what we're going to talk about today with a really terrific panel of speakers. I'm so pleased that our four panelists could join us and give their time to this really important communication, I think, for Rhode Island Bio today. What I'm going to do is start off by asking each of the panelists to introduce themselves, just a minute or two on their background and what their current role is in vaccine development and distribution. And then we're going to talk about what they've been doing over the last 12 months to help end this awful pandemic that we've been going through. So maybe we can start with Michael. I'll probably say your name wrong, Michael. I'll try my Italian on you. Michael Princiotta, how's that?
Yes, that's very good, actually. Mike Princiotta, or Princiotta in Italian. And I'm the CSO for EpiVax Therapeutics. Very briefly, I've been with EpiVax Therapeutics, previously EpiVax Oncology, for about 2 years. Prior to that, I was with Advaxis Immunotherapies for about 4.5 years, working on a personalized cancer vaccine there. And prior to that, I was in academics at SUNY Upstate in New York. And prior to that, I did my postdoc at NIAID, NIH in Bethesda, Maryland. And my focus is on T-cell immunology, with an emphasis on the presentation of antigens and how that affects cell-mediated responses to infectious diseases and cancer.
Great, thank you, Michael. We're going to ask you to talk more about that in a moment, the T-cell aspect of COVID-19 vaccines. But let's first ask Katie Porter also from EpiVax to introduce herself.
Sure. Thank you very much for having me, Carol and Michael. My name's Katie Porter. I'm the Business Development Manager at EpiVax. I've been here for about 4.5 years. Previously in my career, I was with Amgen here in Rhode Island for 13 years. And I was in a very different capacity. I was really on the bench. So I was in quality control and quality assurance and many different roles there over that time, but then made a big change to business development at EpiVax, and it's been wonderful. So I'm very happy to be here.
Thank you, Katie. Thanks for attending today. Next, we'll go to Advit Budkar from Pfizer.
Thank you, Michael. Good morning, good afternoon to everyone. My name is Advait Badkar. I'm a senior director at Pfizer and lead a group called as a Drug Product Design and Development. I've been with Pfizer for 19, going on 20 years now. I have my degree, my PhD in pharmaceutical sciences, and I'm also a pharmacist by training. I have in my career focused on formulation, process development, and new technology for drug delivery, mainly for biologic-based molecules. As part of the Pfizer COVID vaccine team, my group leads is responsible for all of the formulation, process development, clinical supplies, commercial supplies, processes for the lipid nanoparticles that we are all familiar with by now. It has become a household name. And all of the mRNA stabilization technologies that we've worked on. And it's really an honor and a privilege to join you all and represent the great work that has been done globally, not only by the Pfizer scientists, but also by our collaborators BioNTech, based out of Germany. So thank you very much.
Great, thank you, Advait. Terrific to have you here. And then last but not least, we have Kerry LaPlante from URI. Kerry, would you like to introduce yourself?
Sure, Michael. First, to Carol and you, thank you so much for inviting me here today. It's an exciting group. So, it was interesting listening to Michael and Katie talk about the evolving of their career over the years. I'm in academia and a researcher. I just keep adding titles to the existing one. So I'm a tenured professor of pharmacy, calling in here from the College of Pharmacy on this beautiful day. I'm also an infectious diseases pharmacist and researcher at the Providence VA. Why I'm here today, because I've spent pretty much the past, 12 months, not only assisting with the treatment of patients with COVID, but I serve on the state's COVID-19 vaccine subcommittee, which we convened in October. I'll share a little bit of that story later on, but truly an honor to be here today.
Thank you, Kerry. Great to have you. So just over 12 months ago, we started hearing about this new infectious disease emerging from a city in China that I had not heard of at the time. And one of the first things that was done by Chinese scientists and others around the world is that the genetic sequence of the virus was uncovered. The virus was sequenced essentially. And I'm not a vaccine developer, but I understand that this is an important first step in the process of discovering a vaccine. And so I want to start now with EpiVax and particularly with Katie Porter and ask you, Katie, if you can tell the audience what EpiVax did with this sequence and how is it that EpiVax can add to the knowledge in this early phase of vaccine discovery?
Sure, I'd be happy to. So if you're not familiar with EpiVax, we are a Rhode Island-based company. We are located actually in the Rising Sun building in Oneyville, and we have been a Rhode Island company for over 20 years now. We were started by Dr. Annie DeGroot, who began her work in vaccines, specifically HIV and tuberculosis. And in doing so, she discovered a technology where we can screen protein sequences to determine how the human immune system will respond to those proteins, specifically called immunogenicity. And so Annie was looking at pathogenic proteins, finding the immunogenic regions of those with the intent of developing a vaccine with those portions that are most immunogenic, so the body would respond to, but this also makes them safe and effective. And so we did that work, Annie did that work for quite some time, but then biologics, came on the scene, and with companies like Amgen, and the business expanded into biologics where we would identify those immunogenic regions, but remove them. So we've been a very successful business for quite some time because our tools are applicable on both sides of the industry. But specifically with vaccines, as Michael said, as soon as the virus was sequenced, we took the protein sequences of the virus and we screened them through our immunogenicity screening tools, which are computational algorithmic tools. And what they do is they can screen for how the human immune system will respond, as I said, but we cover over 95% of the population. And so in doing so, you're looking for these immunogenic regions that large populations will respond to and mount an immune response. So you're developing a vaccine that is broadly applicable across the population. So we took the sequences from the coronavirus and we found these immunogenic regions. They are short peptide sequences, so we're breaking them down into smaller portions. And then with the intent of sort of delivering a pool of these peptides as a vaccine. And we have since, we've identified those regions, we've since tested them in vitro. And this is really, Michael, how we go about doing vaccine research here at EpiVax. So if you want to think about, the research and development of vaccines, the R&D, EpiVax is the R, and then our spin-off company, EpiVax Therapeutics, is the D. So we're doing a lot of preclinical work now, but the actual vaccine design, the formulation, that part will be transferred to EpiVax Therapeutics, where they will take the product to market. So that's kind of how we started the process. Is there anything else you wanted me to touch on?
Yes, so that's great. I understand that, we're going to talk to Michael in a moment about EpiVax's own vaccine program, or EpiVax Therapeutics, perhaps I should say. But before we get there, can you tell me, do you make this in silico tool available to collaborators? And have you been able to do that in the case of this vaccine or perhaps others?
Absolutely. So we have a couple of different computational tools. The bulk of what we do is in biologics, as I mentioned. We have a tool that many large pharma lease access to. So it's a web-based tool where they can run their biologics to test for this immunogenicity as a key quality attribute of a product candidate. And our clients include Merck, Pfizer, BMS, the list goes on. We're actually getting more and more every day. And so that's the biologic side of things. But on the other side, we have a tool called RI-INBRE, and it does the same thing, but it does have capabilities of actually computationally designing a vaccine. So you can identify these regions, you can string them together into an actual sort of a protein sequence, a longer sequence that contains these short peptides. So that tool really allows you to design vaccines or the immunogenic or antigenic region of a vaccine. And that tool has been used by people including Institut Pasteur in France. And so we do have a number of clients that have access to the tools, but we also offer this service to clients. So if people come to us and want us to look at specific proteins, we can do that and provide an interpretation of the data and a report. And so, yeah, we have had quite a few clients who have access to our tools over all this time, and we continue to add to that list. We're very proud of that.
And Katie, one more thing. You know, we have a mixed audience today. I think some of the audience has a scientific background and others may have more of a business or other backgrounds. So just so everyone understands the importance of this, how would I, as a patient receiving a vaccine, benefit from this careful selection work that you do early on? What is it about the vaccine that I would receive that is better as a result of that careful peptide selection work?
Sure. I think Michael can talk more about sort of the immunology behind it. But as I said, the intent of doing this analysis is to identify the most immunogenic regions and deliver those as a vaccine, eliminating those other regions that really have maybe no value to being in the vaccine. Or there are regions of proteins that can be immune modulatory. So instead of mounting an immune response, they could downregulate an immune response, which would actually make your vaccine ineffective. So we weed out these parts of the vaccine that are most effective, and then also by just using those, it becomes a safer vaccine as well. And Michael can speak to the T cell response and why that actual response is very important. But really, our tools enable safe and effective vaccine design.
So what you do right at the beginning here, just based on the sequence of the virus, could result months later, perhaps years later, in a higher degree of protection, say, for the patient who receives the vaccine.
Absolutely, absolutely. And some of the vaccines are actually based off maybe just the spike protein, which people are probably familiar with by now. But our vaccine, we were actually able to look at many proteins that make up the virus and get those immunogenic regions and deliver a pool of those. So we are actually providing protection against more than, or immune response against more than just the spike protein. And that actually comes into play when we talk about variants. And so we do have coverage over many of the variants that are presenting themselves. And that does have to do with the fact that we don't just rely on one protein, like the spike protein. We actually look at all of them and then pull those regions out that would be safe and effective and use those as the vaccine.
Great. Well, thank you, Katie. Actually, we might come back to the variants later on because I get the sense that we've made a lot of progress in the various vaccine programs against COVID-19. But I also get the sense that the virus is changing too. And that's evolution happening in front of our very eyes.
Yes.
It isn't going to stop, and we may be needing your in silico tools again.
Yes, Michael, it's actually something that we have done a lot of research on. It's called immune camouflage. So viruses do have a way of modifying themselves so that they evade the immune response, which is something we want to try and get ahead of.
Right, so viruses as sneaky little fellows. Okay, let's go to Michael. And I think some of the learnings that you've been talking about, Katie, were passed on to Michael and EpiVax Therapeutics. Michael, what have you been up to?
That's a very good question. So let me just circle back around just very quickly to continue with the theme of what Katie was talking about, because it's very relevant to what we were actually doing at EpiVax Therapeutics. And you're absolutely right. It's not just a sneaky little virus. It's an insidious little animal. And even on Sunday night, Tony Fauci was talking about just what a tricky virus this is in terms of the asymptomatic spread, which is why it's been such a problem, because you can get it, not have symptoms and pass it on to other people. It's very difficult to do population control with something like that, and we're not used to it. Most viruses are symptomatic around the time you become infectious. But as Katie mentioned, we're focusing on a T cell based vaccine, and Katie did a great job of covering, what EpiVax does, how it identifies the antigenic peptides and so on. Just to mention, without diving too deeply into the biology of this, but to understand why we're targeting T cell responses specifically, all of the vaccines that are out there, and they've had remarkable efficacy at controlling disease so far. The BioNTech developed one, the Moderna developed one, and others. But they all work by generating antibody-based responses, humoral immune responses that have to bind to the spike protein in certain places to prevent entry, which is how most antibody-mediated immunity works. So the bug can't get into a cell to replicate. And the issue with this, of course, is that you will eventually get, if the virus is capable of mutating, it'll eventually mutate to the point where those antibodies are less effective or perhaps not effective at all. And this is where T cell immunology comes into play. And the T cells play a couple of different roles. Now, T cells can see, for an antibody to work, it's got to see something that's on the surface of the virus. Whereas what T cells do is they see little pieces of proteins that are derived and translated from the genome of the virus. But that occurs when those proteins are made inside a cell and then they are presented on the surface of the cell. So a T cell can come in and see an infected cell. So it doesn't stop a virus from getting in. A T cell cannot see a viral particle. It can see a cell that has virus in it. But where that is effective is now it can see peptides derived from almost any protein in the virus, which is what Katie was referring to. And it can kill that cell prior to the virus making new viral particles that go on to infect other cells. So it's a part of the immune process that takes place in any viral infection. It's just a different aspect of it. And this is important for a couple of reasons. One of them is there are aspects to the coronavirus that are related to its transmission. And I say this with the caveat across the board that we're still learning about this virus. So none of these things are, written in stone at the moment. But it appears that control of transmission is related to the immune response in the upper respiratory tract. And that seems to be more of a T cell mediated immunity, whereas protection from severe disease or even disease at all seems to be more related to the antibody response in the lungs, in the deeper lung tissue. Okay, so T cells may be very important in preventing spread, number one. And the other aspect of the T cell response is, as Katie mentioned, variants, which is going to be huge, appears to be hugely important with this particular virus. Viruses mutate. This virus is no different. It's so easily transmissible that when you get a variant, it seems to spread throughout the population fairly quickly. With the B117 variant from the UK becoming, I don't want to say prevalent, but quite common in the United States, for example, the Brazilian variant, the South African variant, and so on. And the ability to target peptides that come from proteins that are not subject to that antibody-mediated immune pressure, which is causing these variants. And these variants are in the spike protein where the antibody binds, which is how they're escaping the immune response. T cells can target peptides that are coming from areas in the proteome, if you will, of the virus that are not subject to that kind of immune pressure. So you will likely get a better conservation of response across the variants. Okay, and that's really critical in terms of long-term memory. And I will point out that, for example, there's not a lot of numbers related to some of these variants, but there was a paper just published on the AstraZeneca Oxford vaccine that showed against the South African variant specifically, it's only got 10.4% efficacy, and that's frightening. And the other is something called, and again, without getting too deep into the biology of it, but there's a phenomenon called original antigenic sin, and this is seen commonly with viruses, which is you have enough antibody to recognize made against the original virus, but then you get a variant come in and the antibody responds to that virus just well enough to prevent your immune system from developing a response against the new targets, but not well enough to protect you from disease. And this is actually quite common. So in other words, if you've gotten a vaccine or you've been exposed to the virus, you have an antibody response to that virus, and then you get a variant, and your response is strong enough that you never develop a response against the proteins that have mutated, if you will. So it basically prevents you from developing an immune response against it, an antibody response. This is where T cell immunity comes in. So these are really important aspects to long-term immunity to this virus, and the reason we're going after T cells.
So Michael, you've touched on a number of interesting topics here, but there's one that I just want to pick up on just briefly. My understanding is that there are very few examples of infectious diseases, endemic infectious diseases, that we have ever successfully eliminated. Possibly smallpox was one. I think polio came close, but there are not many examples of this. And I've heard it said that given how widespread COVID-19 now is, it's unlikely that we would be able to eliminate this virus entirely. But if we had any chance of doing that, I think what you're saying is we would need a vaccine that generated a good T cell response. Is that correct?
I think it requires both because of the ability of this virus to mutate. So yes, smallpox is the only one that's been completely eliminated. We got very close with polio, but for actually political reasons, in remote areas, there was resistance to the vaccine. But there are a couple of aspects of eliminating a virus from the population. One of them is that vaccine, the variola virus that causes smallpox, it's a DNA virus. It's a very large one, about 256 different genes or proteins, and it's not very variable, unlike RNA-based viruses like coronaviruses, which mutate much more readily, not as rapidly as say HIV, but much more rapidly. So that's one aspect where it's going to be much more difficult to eliminate. And probably the more important one is polio, smallpox, and for example, measles. These are human-only reservoir viruses. They have no other reservoir. So if you eliminate them from the human population, they have nowhere else to go. Most viruses are zoonotic. They have an animal host of some kind: flu, birds, pigs. In the case of the coronaviruses, bats, pangolins, you name it, possibly cats. You know, it's always got a host reservoir that it can retreat to, or that you can get another version of the coronavirus, which is what happened with SARS-2 versus SARS-1. So, so you have to have the armory, if you will, available for variants or new strains coming out. And this is the other thing with T cells, is that there's a lot of conservation in T cell epitopes between the different coronaviruses. And I'm talking about MERS, SARS-1, the current SARS-2 virus, and so on. So you might get somewhat broader protection as well with a very efficacious T cell vaccine in terms of over time, as you're talking about, in controlling this. So.
Michael, we're going to have a second Rhode Island Innovator panel just on T cell immunity, and we're going to invite you to come and talk on that, because this is a fascinating topic. Anyway, for the immunologists in the room, it is. But for now, I wonder if I can ask you to move on to talk about where you are in vaccine development at EpiVax Therapeutics.
Right, because of the nature of how this is being done, as Katie mentioned, we have short peptide fragments. So that requires making numerous 20, 25 or more different peptide fragments to develop and then pool into your formulation. So in a sense, this is a challenge in terms of commercialization, as opposed to making one specific protein. So we actually have peptides made, being formulated currently, and we're in the preparation of an IND to get into a phase 1 study with this vaccine formulation using peptide with an adjuvant, a more traditional peptide adjuvant type formulation. We are doing intradermal delivery because that's where the dendritic cells, which are the cells that really prime the immune system and a T cell response, are. And we are planning to move into a phase 1 quite soon to show safety. Of course, going to be too small to show efficacy, but to show safety and to demonstrate we get T cell responses. But I do want to mention that we are also currently in a collaboration with another company for development of this formulation to be delivered by microneedle patch. And this is really important in terms of scale up and commercialization and availability, because formulated into a microneedle patch, which is just, it delivers to the same place as it's an intradermal delivery. So you just put the patch on. So these are designed to be actually self-applicable or very easily applicable in a pharmacy, for example, or a clinic, or almost anywhere. And it's ease of administration, but more importantly, the typical patch formulation for peptides and proteins doesn't really have a cold chain issue. These are the way they're packaged under nitrogen, whatever, they're sealed, and they have generally 3 to 5 years stability, and that's at room temperature. So distribution is greatly facilitated, and we all know that the cold chain issue is a real issue with these. So we are actively pursuing this approach.
This is fantastic to see this development work happening here in Rhode Island, and with collaborations with outside companies too. This is absolutely what Rhode Island Bio is doing its best to promote and help develop the ecosystem, so we see even more of this. Michael, we're going to have to move on, but as I say, I would love to talk to you more about all of these things, and we'll do that. Let's move on to Advait at Pfizer. And Advait, I'd like you to tell us about what you've been doing to help bring the Pfizer vaccine to patients.
Thanks, Michael. And this was really interesting to hear Katie and Michael talk about their experiences. So I can remember about a year back, it was March 17th, 2020, when I received an email saying that we have, in principle, agreed with BioNTech to collaborate on the COVID vaccine. That was also the day after we had asked people to start working from home. So it was it certainly was a new world. It was very different. I didn't have a home office set up, and it was trying to work on my laptop, trying to find all of the information there. But luckily, we had a head start. So as Pfizer, we had decided, or we had already entered into a collaboration with BioNTech in 2018 or 2019 to develop a flu vaccine using their proprietary mRNA technology and Pfizer's vaccine development, vast vaccine development background and capabilities. And so we had been working with them as a co-development activity together to identify various formulations, various different constructs and different types of mRNAs that could be used for the flu vaccine. So when we switched the mode from flu to the COVID vaccine, the team essentially remained the same. We could leverage that 1 + 2 years of experience that some of the folks had, as well as we had been developing nanoparticle capabilities within Pfizer internally for our other therapeutic areas such as oncology and rare diseases as well. So we were able to leverage all of those experts within the company and put together a team that was cross-functional very, very quickly. And the mandate we had from all the way from our CEO down was that no expenses spared. There's no option to fail. Failure is not even a word in our dictionary. And whatever you need, you will get. Just make sure that money or resources are not an issue. Just do whatever you need to make this happen. And we initially started with 4 constructs in parallel. And so typically, when you are developing, I would say, a pre-corona program, right? Back in the day, right? When companies work on a program, you start with a funnel. In a discovery, you start with thousands of molecules, then you pare it down. You do some in vitro screening, you do computational work, in silico screening, and by the end of the day, you narrow it down to 1 or 2 lead candidates that you still try to narrow down and take forward into development, only one of them, because it takes a lot of time and resources and money to develop this. Now we were looking at 4 constructs in parallel. We had 1/10 the time that a normal vaccine would have. And we were asked to drop everything else and just focus on this program, which we did. The challenging part also was that we had to work under the COVID restrictions as well. So we had to make sure that we had safety precautions in place. We had to rethink how we would even work in laboratories. How would we have people work in a setting? What kind of screening tools would we put in place? What kind of safety mechanisms can we put to protect our colleagues who are working in the lab? And so it was a very complete reimagination of how we would work. But together with our colleagues in BeyondTech, we put our heads together. We took the sequence that they had. We had the 4 mRNA sequences. We did a lot of optimization around the formulation, which is the lipid nanoparticles. And we quickly tried to we enabled clinical studies within a month of that deal. We were able to then select a lead candidate in the month of July, which we initiated phase 2 studies with, and then phase 3 studies with. And then, as you know, I would say everything else is history. We were able to successfully prove to the agencies globally that the vaccine works and safe and efficacious. And what we also tried to do was, while all of this development work was happening, we were also building our capabilities for manufacturing. We knew that this was not a problem for a few doses or a few thousand, a few hundreds of thousands of doses. We're talking in terms of billions of doses. Something at this scale for mRNAs was never done before. There was, there were no scale-up capabilities to make lipid nanoparticles at that scale. That didn't even exist. So we had to literally go back to the drawing board. We had to redesign equipment. We had to work with the equipment manufacturers, our suppliers. It was the whole supply chain of all of the pieces that fit into the final vaccine product. It was amazing. The industry, all of these people came together, right? They was very focused. They were all dedicated and determined to make this a success. And we were able to not only develop and make new equipment, qualify them, validate them, install them in all of the various manufacturing facilities globally, and then address the question of stability. And we talked about cold chain. I know Michael referred to that, and you also have had some questions on that. For us, it was really all about the data, right? We were not going to take any risks or any chances with safety and efficacy of the product. We wanted to be aggressive where needed, but we wanted to be conservative where we could in order to maintain the safety and the efficacy of the product. And that is why we decided to stick with a temperature for storage for this vaccine, which is ultra cold. And there are reasons for that. We know for sure that when we store something at that lower temperature, that there's physically no movement at the molecular level. And the vaccine is the most stable it can be at that temperature. We, in parallel, had already initiated studies at other temperatures, the more common freezer temperatures, your more refrigerated cold chain supplies as well. But we didn't have time. By the time we were wanting to launch this, we were running against the clock, and we designed shipping lanes, we designed shipping containers, we designed the whole supply chain. Very innovative supply chain colleagues who worked, across the 2 companies globally, and working with some specific technology suppliers. We designed all of this to enable that ultra cold storage chain and supply chain. Now, we are also in parallel continuing to work on trying to improve that and make it more accessible, make it more patient friendly and user friendly. We recently announced that we now have data to show that we could actually store our vaccine for 2 weeks at -20°C, which is the normal freezer temperatures in most residences and most pharmacies and hospitals. So that will definitely help. And there are programs ongoing right now to look at new and alternate formulations and presentations that can even allow refrigerated storage for much longer time. So, all in all, it has been a very exciting and unusual year, I will say, for a lot of us. But the one thing that has kept us going through these difficult times is the fact that we always believe that science will win, and that if anybody can make a difference, it's us. And it's not only our duty, but it's a responsibility. It's a huge responsibility that we have towards the global population for us to be able to be successful. And so that is where what I've been up to for the past, I would say, 12 and a half months now.
Advait, you've been up to a lot, and you don't even look tired. I'm hugely impressed by that story. I have two questions for you. Could you just remind people how long it usually takes to develop a new vaccine against a new infectious agent?
I would say under normal circumstances, it takes anywhere between 7 to 10 years to do this. And yeah, and this was done in 10 months.
And it sounds like it was done without any compromise on the patient safety side, but with a lot of financial risk. It sounds like all of this parallel activity, including scaling up for manufacturing before you even knew you had a product to manufacture. That's something that I guess only a large pharmaceutical company can do. And so I want to ask you the second question. For the benefit of people who are listening who might be thinking about a career in biotech or pharma, how does it make you feel? How do you feel having been part of this team, having created such a fantastic outcome?
That's a tough question to answer, Michael. It's overwhelming, to say the least. It is hugely satisfying. It is you go to school. I went to school for a long time. I did my pharmacy. I'm a pharmacist. I did my master's in pharmacy, my PhD, and all this while, it was always the hope was that if I could even contribute to something that makes it to the end of the to patients, that would have been a huge accomplishment. And in my 19 years, I've worked on a number of programs, but for one reason or the other, I've actually never worked on something that made it all the way to the end. Either we out licensed it, or it didn't work. It didn't work in a particular clinical trial, or the company decided to divest that area. So to then have the opportunity out of the 19 years that I've been at Pfizer to make that impact in 10 months, I mean, that's completely mind-blowing to me. I would say that if to anyone who's interested in a career in biotech or in pharmaceuticals, you definitely need to have the passion to be able to work in this area, and to not waver in your decisions in spite of failures that you may see in your path. This is a field where you see more failures than you see successes, and you always need to have that optimistic outlook towards that. You learn from every failure, anything that, and you implement that into your next program so that you can make that better. And I think it's a very satisfying field to be in, and the impact you can make on individual lives, it's just unimaginable. I've had so many people send me text messages and emails about, we got the Pfizer vaccine, or we got the Moderna vaccine. And honestly, to me, it doesn't matter what vaccine you've gotten. As long as you've received a vaccine and you are much more protected now than you were before, I think that is a win for the industry. I think that is a win for biotech in general.
Well, thank you for that. Thank you, Advait, to you and all your colleagues for all of this tremendous hard work. And I certainly agree with you. A career in drug discovery, drug development certainly does require optimism, but here's an example of how how valuable it can be. Well, so we've talked about vaccine discovery and development, and Kerry's going to talk to us now about another challenge in making these vaccines useful to the population, and that, I think, is vaccine distribution. Kerry, tell us about your work on the vaccine committee.
Yes, thank you, guys. This has been so interesting. So, where my story really began is in October of last year. I received the phone call, I think it was like a Tuesday morning, from colleagues at the Department of Health. I currently work with them in my antimicrobial resistance world. And they said, Governor Raimondo and the Department of Health has put together this task force and they would like you to serve. Governor Raimondo was going to announce this on Thursday afternoon. So can you let us know what you're thinking? And my immediate response in an already overwhelmed environment with, work and home and family and everything that, we've been dealing with, I was really hesitant. But, in the end, my sense of duty and my pride as a pharmacist, scientist, and, a proud Rhode Islander, not originally from here, from New York originally, but, fall in love with the state, I thought, if I could really help, I will find the time to say yes. And you knew it was going to be a lot of work because our charges, and I remember the governor announcing this that Thursday, is that, there was a sense of concern about this vaccine, if it was going to be safe, if steps were going to be skipped. Are Rhode Islanders even going to want this vaccine? Are we going to have a huge uptake issue? But, the distribution and the operations were one thing. We could scale that up, but if we do all this work, are people going to want this vaccine? So she made the announcement and said that I'm appointing this task force, released the names of all of, my now good colleagues that we've spent almost every other Friday with since October, and said, I'm going to ask them to do a secondary validation for safety and efficacy in addition to what the FDA did. And I thought, well, that's a tall order, right? Re-review everything the FDA did, but took that seriously. Immediately started looking at the dozens of vaccines that were with different scientific platforms, learning a lot. I'll fast forward, many of the conversations before that December 12th date were surrounding uptake. We knew that in Rhode Island, and we were one of the better states for vaccination update, specifically influenza uptake. We won an award a few years ago, but even at peak, only 50 to 60% of Rhode Islanders would get their influenza vaccine. And then we knew that there was a sense of distrust with the government. Unfortunately, some populations distrust with pharma, distrust physicians, distrust pharmacists. We didn't know how we were going to manage that. So most of our conversations were surrounding, the safety, the efficacy, and will Rhode Islanders want this vaccine? December happens, we re-review the data, we vote in a unanimous way. It was impressive, approved on Friday with FDA. Monday morning, my colleagues at Rhode Island Hospital were texting saying they have a shipment. It was amazing how quickly it went. And really blown away over the next few months and weeks, realizing that the demand was this high. I personally never anticipated it. I'm so happy to see it. We were even hoping for a 50% efficacy rate. These were the conversations we had amongst ourselves. So just to go back and share that history and the lens of how we were looking at things, and reflecting on that has been really interesting. So, we've moved along nicely. We had to look at the CDC and how they prioritize the rollout. As you know, we knew there'd only be limited doses in the beginning and we had to prioritize people. That was really tough. The CDC had their recommendations in those early days. We in Rhode Island thought we're going to prioritize persons in long-term care facilities because they were not originally on the CDC's list. We knew that we wanted to protect our most vulnerable and our elderly because a good community does that. We know we wanted access, and we know that we wanted to reopen the economy as a third goal, but how to prioritize those groups within were interesting. And peeking at how other states took that same CDC information and reprioritized phase 1, 2, and 3, we realized of my opinion, there was kind of a switching around, you know, everyone just kind of fit into phase one. And there's that kind of mental connection that I'm phase one, I'm essential, I'm, you know, a prioritized population. And Rhode Island kind of stuck true to phase one, phase two populations, but we were seeing how other states were kind of sneaking in other groups and under that phased 2 ABC. So interesting discussion surrounding that. We stayed the course. There was a lot of public comment. I personally received a lot of emails and texts from, you know, homebound persons, people with ALS, people with diabetes, cancer patients, colleagues who are teachers, all asking why they weren't prioritized. And we just kept going back to our framework, which was, we want to protect our most vulnerable. We want to protect our older adults. We want to protect people who are at greatest risk for dying from this disease and also being admitted to the hospital. And as we rolled out, it was tough at times. It feels like it was just yesterday. We had a new governor on, I think it was March 3rd. Things pretty much stayed the same. He immediately prioritized, as did Biden around the same time, President Biden, the teacher population. Luckily, at the same time, the J&J Janssen vaccine was coming to market, so we were increasing the number of vaccines in Rhode Island. And you know, it went fast, and I you know, can't believe we're almost towards that May 1st date where we hope that 90%, actually by April 19th, 90% of the population has an opportunity to be vaccinated. And, you know, like all of my colleagues here, this all happened in the blink of an eye. So with that, I'll pause. We'll open it up for kind of questions and comments, but that's been my story the past few months.
Hey, thank you, Kerry. I have one question actually before we open it up to the general audience. My understanding is that the pandemic has affected certain socioeconomic groups, certain ethnicities, certain races more than others. Did your campaign sort of try to proactively address that, maybe vaccine fears within certain groups more than others?
Yeah, that's a great question. That's what we spent our first 3 months, you know, making sure that all of the persons on the committee and from diverse populations and different community members, religious groups, you know, we're really bringing to the table the perceptions in the community. And we knew that our high density populations in Rhode Island, which are persons that, you know, have multi-generational houses. They are some essential workers. They couldn't work from home like some of us can. They were, you know, maybe there's healthcare access issues. We knew that they were disproportionately affected by this vaccine, and we knew that we needed to prioritize getting that vaccine to those groups. And it's of my opinion, the Department of Health did an excellent job reaching out to community pharmacies, independent pharmacies, community members, and really distributing that vaccine. And I'll even pause. Some of the conversation was, do we just start opening these big military tents in the beginning and increasing access? But we thought, I don't know if that's going to look right in the beginning. We want this to be in the community. It may be a little slow in the beginning, but you know, if it's at our schools and if it's, you know, from our own, you know, populations and people that we trust, it may be not as fast as we would like it to be, but it would be done right, and we would get the uptake that, you know, is needed so we could have some herd immunity in this state.
Thank you, Kerry. That's really important, I know, and thank you for all of your work in helping to get these vaccines to the people in Rhode Island. So I see we just have a few minutes left, and I'm looking at Carol now, wondering how we'll manage Q&A. Carol, I don't know if you already have a question picked out, or I've been seeing in the chat lots of comments coming through. I think Peter Bonk gets the prize for the most comments, so I would invite Peter to ask a question if he would like, or Carol, you may have a key question that you have too.
I think that's a great idea, Michael. Peter was starting a message link here, so let's start with him and see if he wants to use his original question or a new question, because there's been quite a conversation going on.
Yeah, or if anyone wants to jump in just verbally, that's good too.
Okay, I'll try to keep it short.
There he
there's
is.
obviously a lot of concerns, but my biggest one is, we knew in July or August that a vaccine would be available by the end of the year, or quite certain. It was quite probable. And I would like to know if the CDC, the various states, Rhode Island here, Connecticut, did they game out like how to distribute a vaccine that's going to have high demand but low availability? And we're still in that lower availability, though that is eased. To prioritize with the in my mind, the condition should be minimize deaths and minimize hospitalizations. And what is the best way to do that? We already had 6 or 7 or 8 months of history with the virus. We know who's vulnerable. We know who's not. We know to some degree how it's spread and all that. So were all those factors taken into account? I mean, it just seems like the rollout was kind of haphazard and not well planned out in many cases. The idea that we're going to give it to kids, the vaccine was never approved for kids back then. So we had plenty of time to game this out and plan it. So that was really my concern. I didn't see much of this. There's a question in there, I guess, Carrie, sorry.
That's okay. No, I appreciate your viewpoint on this. So watching it all roll out as you have, Peter, I reflect on this often. It's hard to say because I would get the little pebbles of information as they were coming in, and the strategy of let's bring in CVS and Walgreens and the pharmacies, where I was not a stakeholder as a pharmacist on that committee, and I was I use my role what's the best for all, but admittedly had a disclosed bias that we hear all the time that 90% of Americans are within 5 miles of a pharmacy. And I will say pharmacies here in Rhode Island have provided the most amount of vaccines at around 90,000 as of this morning. So I think getting these, in my opinion, getting these vaccines to the pharmacies, getting the 7 or 8 small community locations, turning over both the field hospitals to become mass vaccination sites, took, I don't even want to say a while, but took some time. I'll kind of pause and say myself as a vaccinator and working at a couple of these clinics, when I sit there and draw up these vaccines and watch the operations, I'm personally taken back from the time the vaccine arrives and we're checking the temperature every hour to unpacking and the gloves and the syringes and which syringes and the alcohol swabs and logging all the different cards and the lot number, and is the lot number match what's on the iPad, and do the appointments and the people coming in. I can go on and on. I take a moment and look around those sites, and I can't believe how much we have accomplished in a short amount of time. So I can only share my viewpoint here. It's a lot, and it's March, and this started 4 months ago. Here in Rhode Island, 20% of our state is vaccinated. We're, as of this morning on the New York Times page, we're a top 5 or 6 state. Sometimes the report card looks great, other days it doesn't. We celebrate when it is. We try to figure out when it isn't. But my last point is, from my standpoint, I'm very impressed on how well this came together. And right now in Rhode Island, 90% of doses each week are getting into the arms of persons. So I think it was the typical curve of getting organized, getting together. You really can only feel that operation when you're in it. I never really knew what it would be like until I started working in these clinics. But now we're there, and I think it's moving forward. But I really appreciate your perspective, Peter.
Carrie, let me follow up on Peter's question. God forbid there was another pandemic next year with another virus. Your task force is still in operation. Are there some things you would do differently second time around, do you think, based on the learnings that you've had?
That's an excellent question. Like I said earlier on, I was so pleased to see the uptake. I didn't know, the demand would be so high in a good number of populations. So I think that time will tell that they'll hopefully continue to be trust. Operations are good. You know, there's nothing I can really think of right now. You know, the teacher conversation and having reopening the economy and protecting our frontline workers, like grocery store employees personally. You know, they've been on the front line since the beginning. They didn't have an opportunity to work home, as our bus drivers. So, you know, hopefully we could just get more vaccine in a quicker amount of time and not have to make those difficult decisions on who are we prioritizing and why. So.
Very good, thank you. I'm just checking my time here, and it looks like we're at the top of the hour, as they say. So I guess Advait and Michael and Katie, you miss out on the questions. Carol, is there some way for them to answer questions after the event based on the chats that have come through?
Well, they can always send their questions directly to me. I believe my email was on the Eventbrite invite. People are welcome to send the questions and we can go back to the group. We also can get the questions right from the chat here. So we can address them that way, but it's amazing how quickly this time went by. We always think we're going to allow enough time for questions, but the conversation was so good we could have gone on for many more minutes. And I want to thank Michael for moderating, and for Advait, Carrie,
thank you,
Katie, and Michael for participating, and all of our sponsors, and to my co-host Bongsup Cho from Rhode Island INBRE. This was a terrific session. And Michael, Elliott, any last words that you want to say to everyone?
Just to echo what you said, I'll have to go back to moderator school so that I can leave more time for questions. But really, I want to thank all four of our panelists for really excellent contributions to the event today, but more importantly, just fantastic contributions to helping to end this awful pandemic. And for those of you who are Rhode Islanders, I think Advait will probably become a Rhode Islander after this event, but the rest of you are Rhode Islanders, and I think we're all very proud of the work that you've done. Thank you very much.
Thank you.
Thank you all.
Great, thank you.
Goodbye, everybody.
Have a good day.